Thank you everyone for joining today. For those of you who don't know me yet, my name is Alex from the Molecular and Genomics Informatics core. Today we have the pleasure of having Twist joining us. We'll be talking about their synthetic biology and custom NGS library preps. But before I turn it over, just the general housekeeping items. If you have any questions, please drop them in the chat or the Q&A box. We'll monitor it and answer what we can on the fly. Anything that's not mentioned during the talk, we'll ask at the end. So with that, I'll kick it over to Alan and Stephen from the Twist team, and let's hear more about the awesome opportunities that Twist has.
Great. Yeah. Thanks everybody for joining. My name's Stephen Weber. I manage the field applications team in the US for the synbio side at Twist. And then Alan, did you want to introduce yourself really quick?
Sure. Yep. So I am Alan Fox. I am a field application scientist that covers New York down to DC. So I cover New Jersey, and I will be covering the NGS portion of the whole catalog. Take it away, Steve.
Thanks. So yeah, Brennan Hicks is your account manager on the synbio side. I'm filling in for him today because he had an emergency dental surgery, but we're making it work. And so I will be joining for the first half of the call. I will go over our synbio portfolio and then take any questions. And then I will be dropping off around the half hour. So I'll go ahead and share my screen now. Can everybody see this okay?
Yes.
Great. So I'll start just by briefly talking about our oligo synthesis technology. So this really is the core foundational technology that Twist has that feeds into all of our different applications. And essentially on a chip with the same footprint as a 96-well plate, we are able to manufacture over 1 million unique DNA oligos. Now, these oligos not only are they very high in throughput, but they're also very high in quality. This quality is measured both by our error rate of being about 1 in 2,000 nucleotides on average, and our uniformity, which ensures that the vast majority of oligo members in a given pool will be roughly the same length, roughly the same molar concentration. So these oligos could be anywhere from 20 to 300 nucleotides in length. They are extracted from the chip in pools. Those pools could be anywhere from, let's say, 100 to hundreds of thousands of members in size. So those oligos are available as a standalone offering, and I'll get into that a little bit more. But the other kind of portfolios you see here are also ways that we use these oligos to help get a little closer to the desired downstream product that might feed directly into one of your preferred workflows. So I'll start on this genes offering. So Twist's genes really allow you to take whatever in silico design you have and turn it into a physical product. Very short lead times, very high quality, and very scalable. So there won't be any effect on the lead time if you do have lots and lots of genes that you want to order. Now, the quality is going to be industry leading for these gene fragments, hovering around 1 in 5,000, 1 in 6,000 base pair error rate. So it'll be very easy for you to find an error-free clone if you do choose to go with the fragment option and clone yourself. Now, there's a number of advantages to going with the Twist offering. There are other gene fragments, gene clones options out there. You'll find that Twist has industry-leading price, but you also don't have to sacrifice quality or lead time for that price. So some high-level information here. The gene fragments can go up to 1.8 kb in length, and you'll get anywhere from 100 nanograms to about 1,000 nanograms with those. And those gene fragments started around 7 cents a base, so industry low pricing for that. And then we also have our clonal genes offering. which can go up to 5 kb insert size. So your synthetic insert can go into a number of different backbones, and you can choose what scale up quantity you want, and get a sequence verified error-free plasmid as a deliverable that should be ready for your downstream workflow. So Twist does have a number of catalog vectors available for you to use. This includes cloning vectors, gateway vectors, lentiviral expression, bacterial expression, and mammalian expression vectors. Now, if what you want to use is not compatible with any of Twist's catalog options, you can always use our custom vector onboarding program to basically send us your plasmid, and then we will go ahead and prepare that for our cloning method. So, of course, there's a number of different applications that could be applied using these, and we're happy to help figure out kind of what makes the most sense for you based off of your budget, workflow, etc. Any questions right now about the genes, fragments, or clones? So I'll move on to our oligo pools. I mentioned those briefly, but here, you'll be able to distinctly define a large number of variants or different sequences that you want to include in a single pool. So, like I said, you could have anywhere from, let's say, hundreds to hundreds of thousands of members or millions of members in your pool, and those could be variants of a certain sequence. They could be different RNAs or CRISPR guides. So you'll have a lot of flexibility of getting very large and very long pools. So really, the sky's the limit as far as how you're going to design these pools. This will be totally user defined. Now there are ways that we can help with design and execution of using these pools with our library group, which I'll get into a little bit more. But here's some ideas of different things that you could potentially code for in a given pool. So why does it matter that the Twist oligos have industry-leading uniformity? Well, this can be very impactful on a screen. So, if you have a high throughput screen and you're testing lots of different mutants or different sequences, then you want to be able to find in your sequencing those different hits. And if you have a large skew of uniformity towards certain variants, those might appear to be hits, where in actuality, those are just a result of the bias of the pool that you're using. So alternative options out there might result in the confounding of your data and less powerful results. Also can just require a lot more sequencing. So the cost of the sequencing could be far greater if you're using a lower quality pool. And then of course, the 1 in 2,000 base error rate is very important for us to be able to deliver up to those 300 nucleotide oligo lengths, which will ensure that the vast majority of those molecules are in fact error-free. So, you really have virtually unlimited scale of the pool size here. So rather than thinking of it like a pond, it's more like an ocean of different possibilities. And then, having that length really enables you to use many different workflows. And here's some examples of different applications where these oligos are used and can be used. Of course, making something like a CRISPR guide RNA library is very easy using our platform. Variable inserts only about 20 nucleotides in length. Also, if you're doing something like a massively parallel reporter assay, you can shuffle reporters, shuffle UTRs, things like that. You can use it for fish or oligo paint, as well as things like making amino acid variant libraries or short other RNAs or peptides. So the workflow usually looks something like this. You design, specifically for CRISPR, you could design your guides, you print a pool, amplify it, clone it into your backbone, and then you make your virus and you perform your screen. Now, Twist can help with not only making the oligos, but we also have recently added our cloned oligo pool offering, which will get you through the oligo pool manufacturing, amplification, cloning, and then we would QC this cloned pool, telling you exactly within that plasmid DNA population how frequently we will see any given variant. So this is very useful for helping to deconvolute any potential skew that you might see in your results. And we are very capable of making these very high diversity and high quality libraries, carrying the uniformity and low error rate into that clonal plasmid population. So another way of looking at here, using basically the same workflow, the oligos will be able to find, you'll be able to find those in your NGS data very easily. So I'm going to move on to our variant libraries offerings. So we have at Twist, using those oligos, a number of different applications. I mentioned kind of that downstream workflow with cloning oligo pools. We can also use these oligos as kind of distinct amino acid variants for a number of different formulations of coding sequence libraries. So using these oligos, we can define not only which amino acids are present, also define the ratios of different amino acids, codon usage. We can control the diversity by avoiding restriction sites and as well as avoiding unwanted amino acid motifs. And then, of course, we would be qualifying all of these using NGS. So Twist's libraries are clearly advantageous to some of the more legacy techniques like degenerate synthesis or trimer synthesis. We really are able to distinctly define every member, and we don't run into any sort of sequence-dependent biases. And this becomes especially important as you increase the number of variable positions, because any off-target or designed unwanted errors will become more and more likely as your pool gets larger. So you can think of this rather than when you're screening a library looking for a needle in a haystack, the needle being an on-target desired variant. When you're using the Twist library, you are looking for a needle in a stack of needles. So really effectively reducing or increasing the effective library size, making sure that you can find what you need in there. So again, there's a lot that you can't do. Oops, sorry. A lot that you cannot do with a more traditional library approach. And the Twist method is really going to help you to find what you need quickly and effectively, thus saving a lot on your downstream costs. So if you're familiar with degenerate synthesis, there's a method commonly referred to as NNK, which stands for about 32 of the 64 possible codons. And this includes at least one copy of each of the 20 amino acids. Now, you would expect if you have 32 different variants in your degenerate library to get kind of even representation of those 32. Well, in reality, there is a bias, most notably towards the G nucleotide, that creates kind of a big mess of different variants, which makes it very difficult to find some of those underrepresented ones. And it will be very predictable in each variant that you're going to see kind of certain amino acids coming up, as many as 10%, others as low as 2% of relative abundance in that pool. Versus the Twist library, we're really able to very accurately achieve very, very balanced diversity. Again, starting with exactly the variants that you want, avoiding things like stop codons, which are going to be present in a degenerate library, as well as avoiding all of this unwanted skew. So this technology can be used for a number of different kind of chemistries. The most basic of those is a single site library where you're just changing one amino acid position at a time. So this is commonly referred to as a scanning library or a deep mutational scan. And we have a way of making this using pools of those distinctly defined oligos that's going to generate exactly the diversity you want. And then we also have a number of different combinatorial options. Both options 2 and 3 here are kind of different formulations. You can look at this. So essentially, one of the options is kind of having a little bit of diversity scattered across your gene, and the other one is having lots of diversity packed into one or more confined variable domains. So I'll get into a little bit more about those. So the first option, the single site library, essentially you're going to have the ability to define exactly which amino acids and which codons are present at each one of those positions. This allows you to do things like choosing all 19 non-wild type variant amino acids. So we can even eliminate wild type from there to help increase the chances of you finding all of those different mutants. We could even do things like single codon deletions. And essentially, once you tell us which positions you want to have mutated and what you want them to be mutated to, we will print small pools of oligos, dish them out into wells of 96 well plates, however many wells you have amino acid positions you want to vary. And in each one of those wells, we would change a single amino acid to each and every different variant that you want using a basic site-directed mutagenesis reaction. Once we complete this reaction, we'll be left with double-stranded DNA fragments that contain whatever flanks that you want for your purposes of cloning. And we'll do NGS QC to confirm the relative abundance of variants at each one of those positions. Now, we also can perform cloning of that library fragment into the vector of your choice, followed by another round of NGS QC. Now, I'm going to talk a little bit about our combinatorial library offerings. So we can using the same kind of one oligo per variant kind of idea, we can generate very high diversity libraries, mutating multiple regions or domains to whatever amino acid profiles you might want, achieving up to greater than 10 billion total variants. Now, most simply, you can think of these types of libraries as printing pools of oligos that will code for variants in one or more of these stretches. And then we would be stringing those together, shuffling them, and generating a library fragment that would contain all of the combinations of variants within and across each one of these variable regions. Here we're using an antibody domain as an example. This is a very common application for these kind of domain style libraries, most notably because these antibodies have CDRs, which are hypervariable regions, where the rest of it is mostly constant. So it's a perfect fit for this type of approach where we can engineer exactly the diversity you want into each one of these sections. Also, one of the known limitations of synthetic libraries for things like antibody discovery is that traditionally they would have unwanted sequence liabilities present. This is the case with both degenerate or a trimer-based technology, essentially things like glycosylation sites and deamidation, these di and tri amino acid motifs that will cause issues with manufacturability. We can essentially remove those in silico from the library to begin with, so you don't have to worry about screening through that junk. Now, within each of those variable domains, you'll be able to choose, again, not only which amino acids are present, but also their relative abundances. So here's an example. Essentially, you'll have the ability to enter whatever percent of each. In this example, we're kind of biasing towards wild type a little bit to limit the number of total changes on average, while also over or under representing certain variants. And here we're showing off that we're able to achieve very accurate diversity down to as low as 1% abundance. Now, another way that we can engineer these is if you actually had distinct sequences in each one of these sections, you could actually give us, almost like pools of oligos, and then have us shuffle these together for you and then assemble them. So one of the potential limitations of having kind of this one oligo per variant idea is that if you have a region of consecutive diversity, in this example, 8 positions that have a total of 5 billion possible variants, we would never, we can print 1 million oligos, no problem, but what about 5 billion? Oh, probably not. So at this point, we went to the drawing board and we found a method that we could use that will assemble various pools together without any sort of constant positions in between them. This way we can achieve the exact diversity profile with all of the requirements, while only using, let's say, tens of thousands of oligos to achieve billions or more diversity. We can also use this technology to very efficiently introduce length variation into a pool. So really, the sky's the limit as far as how you design these, and Twist will likely have a good solution for any sort of library design you have. Now, lastly, I just want to mention we have another kind of combinatorial technology. Differs slightly from the domain style libraries, which require kind of these variable sections with then constant regions in between them. This spread out low diversity or SOLD library allows us to essentially eliminate the need for any sort of constant positions at any place in the gene, and we can this way introduce a little bit of diversity across a larger stretch. So this is most notably useful for things like enzyme engineering, where maybe you did perform a single site saturation, or maybe you're using crystallography data and you found that your most interesting sites that you would like to then shuffle together and see what happens if you change combinations of positions if they're not co-located in your linear DNA. So essentially, using this technology, we can introduce diversity we can qualify them in chunks using NGS, and we find that the uniformity across each of those chunks is very even, one being the normalized average. And you can see the fewer variants there are, the better the diversity is. So it's kind of the inverse of the other type of library where you can pack as much variation you want into small confined regions. Here you can pack a little bit of diversity anywhere you want. So between the two, virtually any library design will be possible. And of course, I mentioned we also can clone these libraries for you, so we can deliver plasmid material that will be ready for your downstream use. So those are all my slides. Do we have any questions?
I've got a quick one if no one else has one, just for a fun one I'd like to ask whenever we're talking about synthetic biology. What's kind of the largest vector or full size fragment you guys have synthesized so far?
Yeah, so with our genes offering, we are currently offering early access up to 10 KB inserts.
Okay.
So that's kind of the farthest that we've pushed it in a commercial setting. I'm sure the R&D team has done some crazy stuff that maybe is not ready for prime time, but yeah. And then as far as a vector backbone, probably up to 10 or 15 KB. So potentially one of those constructs could exceed 20 KB in theory. So
awesome.
Yeah, if there are some crazy wild projects out there, definitely bring them to us. We're happy to take a look. And even if you think it exceeds our standard requirements, Twist is always pushing forward on the bleeding edge of synthetic DNA, and hopefully will help to make those hard projects happen.
Cool. Anybody else have any questions for the synthetic biology part? Or else we'll switch over to Alan with the next-gen sequencing.
All right. Well, in that case, I'll pass it on to Alan. We can provide the slides that I showed you today, and maybe send them out to all the viewers. I'll coordinate with Alan on that. But I just want to thank everybody for your time. Enjoy the rest of the presentation. And if there is any interest in talking about some of these options on a more one-on-one setting, if you have any upcoming projects, we encourage you to chat with us. You'll get on the phone with myself or a member of the field applications team, and we can chat through your projects and find a good solution.
Cool. Well, thank you, Stephen.
Thank you. Have a good one, everybody.
You too.
Bye.
All right, Alan, ready for some NGS?
Yes. Awesome. Yeah, so Stephen gave you a great overview of our synbio offering, and I'm going to jump to now our next-gen sequencing arm. So, here at Twist, we make DNA. So like Stephen mentioned, we make oligos. We make really high quality oligos as well. So what do we use that then on the next-gen sequencing side? Well, we use that to make capture panels or probes or baits, however you want to call it, and also making controls. So our full breadth and offering that Twist offers is, the synbio portion, like Stephen mentioned, with genes, oligos, and libraries. I'm just going to talk to you about the NGS, but we do even have an ARMS for in the biopharma space, like Stephen was mentioning with our libraries, where you can eventually make some antibodies. And then we even have eventual, as we grow, making more and more DNA in the data storage space, which is going to be really cool in the long run. Just think about, right now, you're storing, everything on a hard drive and essentially zeros and ones. Imagine if you can do it in A, C, G, and T. And, if you need to replicate your data, you just do PCR. If you need to read it, you sequence it. So it'll be pretty cool in the long run, but let's just talk about NGS for today. So just a quick mentioning of what we offer in the synthetic control space, and then we'll jump to custom probes and all that. So one thing at Twist, we offer some viral controls, especially in the respiratory side, where we have influenza controls, some rhinovirus controls, coronavirus, SARS controls as well, where we even kind of went towards the pandemic side of things, seeing that there's a huge need into just how do you go about QCing an assay, or, if you're doing qPCR, if you're doing our Twist enrichment assays, you need a positive control. So, having all these different types of assay controls, essentially, even for monkeypox, which we just recently released, or even Omicron or anything related with SARS, we can help you with those needs. But then we also went into making a cell-free DNA reference control as well, where taking this whole idea that we can make, really good high-quality synthetic DNA at various lengths if needed. But what we decided to do is actually, well, let's just take make synthetic DNA, make it essentially about 160 to 170 base pairs to mimic cell-free DNA, and then spike that into a background of cell-free DNA that is donor-derived. So now you have essentially a control that we control the variants. So we have a bunch of nucleotide variants, so S and Vs, some indels in there, deletions, insertions, as well as structural variants, and put them at various degrees of allele frequencies from 0%, which is essentially just the wild type background donor DNA, to 0.1, 0.25, 0.51, 2, and 5%. And then we actually control and test the quality of this using NGS to make sure that all the sites that we put in there and made synthetically are there. And then we even digitally droplet PCR it for a subset of the alleles and sites for the lower level, the 0.1, the 0.25, and the 0.5 allele frequency controls. So what we did is just make essentially a really good high quality control for your needs that you could utilize for any cell-free DNA or liquid biopsy assay that you're doing. But jumping to more the NGS side of things. So here at Twist, we offer the ability to do library prep and target enrichment. So you extract your DNA, you extract your RNA, and then you're going to jump to us for library prep or target enrichment. And then after you do that, you'll do sequencing on Illumina platform or PacBio or Nanoport, however you go about doing it, and do your analysis. But what we can help you with, we can sell you multiple levels of library prep. So we have the option for if you want to use our enzymatic frag kit, which actually works really well on FFPE samples, where you essentially just use a series of enzymes to chop up your DNA into smaller pieces. And then, or a mechanical frag kit where you're using a Cavaris or a Megaruptor. If you're going to do long read capture, you use a mechanical frag kit because you're going to get really big size, 6 kb, 5 kb, 10 kb size fragments. But however you go about the fragmentation method, we are here to help support you in that sense of things. And then we provide you multiple different types of adapters as well. Whatever, pick you and choose how you want to go about doing it. Our combinatorial dual index adapters are a full length adapters where once you ligate it onto any piece of DNA, you now have the ability to cluster onto an Illumina flow cell, as well as the sample barcodes to be able to tell up to 96 samples apart. Or a universal dual index adapter system, which uses a short stubby adapter, just a small one that ligates the same regardless of what sample you use on both sides. And then through PCR adds the ability to add the sample barcodes and the ability to cluster on an Illumina sequencer as well. And we have currently up to 384 combinations of that, but it is expanding to even higher amounts pretty soon. And then we even have our UMI adapter, so a unique molecular index adapter. So for anyone who wants to do like duplex sequencing or go really low level allele frequencies and using like our cell-free DNA reference control, that's what you would like to use. It works in the same system as our UDI system, such that it's essentially a short stubby adapter with UMIs a part of them. And then you add through PCR, again, the sample barcodes and the ability to cluster on the sequencer. And we can provide you even the purification beads, as well as we have a really good hi-fi PCR master mix, which is just as good, if not better than Kappa. So, highly recommend trying that out. And then on the target enrichment side, we offer 2 different flavors as well, depending on kind of what assay you're trying to do or how fast or slow you want to go. So we have our standard HIBE reagents and wash buffers, which essentially is an overnight HIBE. So you would do the library prep and then set up the HIBE and then have the HIBE go about 16 hours to 18 hours overnight, and then do the enrichment steps the next day and put it on the sequencer. Or we offer a fast HIBE option as well, where if you want to do the whole library prep and do the enrichment and the target capture and everything all in one day, we have that ability as well. And then we can offer you a bunch of fixed panels, which I'll go to in a little bit, or custom panels, which is kind of the other highlight of this talk. Whatever you want, we can take whatever you need and make a panel for you. And then we provide you everything you need, including the striped adamant beads, the DNA purification beads, blockers, and even a polymerase as well. So provide you everything that you need to do your library prep and target enrichment, whether you're doing short read or long read. And just to kind of mention some of the fixed panels that we have out there. So let's say, you don't want to deal with making a custom panel. You just want to kind of use something that we have off the shelf. We have a bunch of options for you. I'm just kind of highlighting the few of our top ones that we actually sell. So we have multiple flavors of exome, but our top ones are like our comprehensive exome, which is our legacy exome. It's lean, it's basic exome, great performance. But then we tweaked it a little bit and came out with our exome version 2 to then have it have a little bit more clinical relevant mutations in it. So we have some like clinically relevant SNPs that are outside of the exome. We cover the TERT promoter, things along those lines. And we actually adjusted to make sure the stats and the content would work great on a NovaSeq. So it's one of the, if not the best in class content and performance exome out there on the market right now. We also offer a bunch of viral panels as well, if you need any. So obviously we have the viral controls, but then we have also viral panels as well. So we have a comprehensive viral panel, which has like 3,000 viruses on there, DNA, RNA viruses. But then we also have a respiratory viral panel, which kind of is more, like the 13 virus controls that I mentioned, more focused on those regions. So like influenza, rhinovirus, things along those lines. And it's actually heavily used even because for SARS as well, where we even have a SARS specific panel if need be. We have a good offerings right now with their methylation. I'm going to go into it a little bit, but not very much. So if you want any more information, happy to set up a call later. But we have our methylome, which covers about 4 million CpGs across the whole genome, and is kind of a way to hopefully replace using an epic array or microarray, which only has 850,000 spots. If you want to go bigger, get single base resolution, and get better data realistically, definitely recommend reaching out to us about the methylome. And then we do have, well, if you just don't want to go so big, because 4 million CpGs is pretty big. If you want to just look at smaller amount, only what's related in cancer, we do have a pan-cancer methylation panel as well, which is only like 1.5 megabases in size, so a smaller panel. And then we also have, I didn't put it here, but a mouse and a canine exome as well. So we do have other abilities to make other genomes than just human too. And let's just say you want to take one of our off-the-shelf panels and, maybe it's not to your liking. Maybe you want to do a little bit more to it, add some different content. Well, we have this module-based approach to our system, which is what makes us really stand out above the competition. You can take any of our fixed panels and you can add any content you really want to it as well. So you can take our exome and add a copy number backbone panel to it, or take our exome and add a mitochondria, or take our viral panel and add some bacteria content to it. Things along those lines make like a pan-pathogen panel. So we can really help you in any way, shape, or form to, you can start with a custom panel and then add something to it later. We can really work with you to either make a spike into it or blend it together into one panel, whatever will fit your needs and what you're trying to do. And that kind of brings me to my next point. And what we're trying to talk about today is more customization and going about the whole, well, you have these specific regions of interest, you have these specific bacteria you want to look for, the specific virus, specific exons, whatever it is. This whole design process, we work with you along the way, and I'll go into some of the ways that we go about doing the design concepts in a little bit. But this whole design process, you reach out to us, we'll work with you and start, designing a panel for you, and it's completely free. Usually, if you submit it to us, like this chromosome start and stop, these bacterias, this whatever fusion, whatever it is, this long read capture region, whatever it is, it only usually takes about a day or two to get you, here's what the panel design would look like, completely free of charge. So, it's a good consult. We can just reach out to us, we're happy to help, at least give you an idea now. All right, well, this is how much it will cost to build it, and how much it will cost to actually do, buy all the reagents and things like that as well. And then, you can move forward with that quote. So if you do choose to, purchase everything, it only takes us about 3 weeks to build the panel and, get you everything. And that's much faster than the competition out there, such that then you can take that build, you can test it. And then if there is any tweaks that happen to be needed to be made from this custom panel, which to be honest with you is rare nowadays, it seems I've only had a handful of panels actually ever have to go back through this process again, because maybe you have a probe that is over a repetitive region that should have been removed, or a probe that didn't work as well. We need to add more of it, things on those lines to help the balancing. But very rarely do you have to go back through this whole process. So realistically, you can go from reaching out to us tomorrow to having testing in your hands and having all the data within a month, faster than you could even get a panel from any one of our competitions out there. But now let's go into well, what is some ideas for design? What are you trying to do? Things along those lines, and kind of how we go about doing all these custom designs that we do, because we don't just always deal with human, that's for sure. So, like I mentioned, we don't always deal with human. We can deal with any genome. So we can deal with bacteria, plant species. I've done monkey, mouse, rat. I've seen a ton so far. All you really need to do is just, in those cases, just tell us, all right, well, here's the genome build that I want to use. Here's the FASTA sequence. Here's the bacteria. Here's the viral sequence, whatever it is. Just give us that, and we can design probes against whatever you need to in that sense of things. In the human world, or a mouse, or all that, but mainly human, just let us know. You want HE19, HE38, you want the new telomere to telomere genome build, which I've done a couple. Whatever you need, we're happy to help you start there, but we need to know what genome build we're going to start basing our designing the panel against in our probes. And then we take a lot of all these things into consideration when we're trying to design the panel. Is it diagnostic? Well, are you looking at cancer? Are you looking at microsatellite instability? Because we have a completely different way of designing it. Are you just looking at mainly hotspots for cancer, where it's, just mainly looking at like a SNP or, specific exons? Are you looking at translocations or inversions? That's a completely different way of going about designing the probes, especially if you're doing it on DNA versus RNA, which is my next slide. Are you looking at copy number variation within the gene or outside of the gene across the whole genome? Are you dealing with pseudogenes? Is there something that we have to take into account when designing it as well? Or even big, large indels, things on those lines. And then, if you just say, all right, well, here's just a list of genes. So here's 200 genes, here's 50 genes. Please design against, well, what, the exons? Do you want us to put the UTRs in there? Do you want just the whole entire gene, like spanning even the intronic regions? Do you want the promoter? All that comes into play, even the transcripts. Do you want like all the possible transcripts that are available with the alternative exons? Happy to work with you in that sense of things, but we need that little information just to know, and tailor the panel to what you want. As well as, well, if you just give us coordinates, a lot of the same things comes into play as well. So if you just give us the chromosome start and stop, well, is there anything that we need to consider, like MSI, like I was mentioning? Is there any variability there in the sequence? Any SDRs, so short tandem repeats, that we need to be, aware of, such that we design the panel around that accordingly? Or is it anything difficult? Are we talking HLA, IgG, or, like the TERT promoter, anything where there's a lot of variability or it's just really hard to capture, things on those lines? We've already actually designed a lot of panels around this. We even have an HLA panel coming out eventually. So, happy to work with you in any sense of things to kind of make sure we get the panel right for you. But one of the biggest things that we need to know is, well, what are we designing the panel against too? So are you going to be working with genomic DNA or RNA, and thus have to make it cDNA? And that comes into a lot of times just the idea of, well, how do we go about doing the actual panel design in the sense of exons? So if you give us a list of genes and you're looking at genomic DNA, well, it's perfectly fine. It doesn't matter what size the exon is. If it's, let's say, in this case, greater than 120 base pairs, like 240, and we could design our 120 base pair probes, one right after the other, no overlap at all across this exon, completely, fully spanning it, regardless if it's genomic DNA or RNA based. But if it is, let's say, an exon smaller than 120 base pairs, well, if we're designing it against genomic DNA, well, then we're going to have this going, the 120 base pair probe going into the intronic region, or even, the UTR or promoter, depending on where this, sequence lies. This probe won't really work very well, especially if it's only 50 base pair exon. This probe won't really work very well on the RNA side. So we kind of need to know those logistics when we're trying to go about doing it. And I kind of mentioned here with cDNA is, we could design, for both DNA and RNA, but you need to, for the RNA side, our library preps are DNA based at the moment until we eventually come out with an RNA-seq kind of library prep kit. But actually, a lot of people really like that our library prep's only DNA based, because you could take any RNA, put it through, let's say, superscript or NEB's reverse transcription modules, make it double stranded, and then just put it through the same workflow you would with DNA. And when you do this, if you extract total nucleic acid, you have then DNA and then eventually double-stranded cDNA all in the same actual library prep. And then you can take that library and you can split it into a hive for RNA and a hive for DNA. So that way you just do one library prep for both, DNA and RNA in one tube, especially if you have total nucleic acid there, and split it however you want. And a lot of people really like that flexibility where it's not like I only have to make an RNA library prep or I only have to make a DNA library prep. At least you're doing, the same thing all at once. You just have to do the reverse transcription ahead of time. And the other good thing too with this as well is, let's say you want to not use our library prep and use like 10x single cell, whatever, RNA-seq library prep that 10x offers. You can still go put that into our hybridization system as well. So we've seen a lot of designs against like 10x, just trying to pull out those alternative transcripts and all that as well, because you have to see it's a really high depth to really pull all that out. So if you only care about X the number of transcripts, using a target capture based approach, it's a good way of going. So we can take, again, this will come into play whenever we're designing the actual panel. And if you're doing the other note, if you're doing RNA, we're going to make sure that we at least have a probe that will span an exon to exon junction and not deal with the possible of an intronic region as well. So that's kind of why it's really good for us to know DNA, RNA based, how you're going about doing the library prep, things like that. And that's where I can come in, help talk to you, through it as well. And one of the things too about our probes is, let's just say, all right, I'm submitting, these regions of interest. I want, X amount of diversity there, or I'm looking at these bacteria or these viruses or these fungi with all these different species, all these different strains. I want to make sure that my probes are going to be able to pull out the regions of interest, or at least the strains that I'm looking for. Well, we have many ways that we could think about dealing with the probe design, especially when it comes to, are you pulling out an oncology sequence, a cancer mutation event versus, the wild type, and will the probes still be able to enrich for it? Well, the good thing is with our probes is they have this randomly dispersed, discontinuous mismatches built into it that it can handle up to 10 to realistically about 15 before it kind of starts breaking down. If there's continuous mutations across the whole probe, so it's 120 base pairs in length, let's just say, if you look here, it's pretty, it drops in the efficiency as you go pretty far down, but let's just call it at, let's 60% about here with 30. You still get 90 base pairs of our probe still working. Even if 30 base pairs of it doesn't work on either end, the 90 base pairs will still be able to enrich and still pull out the sequence that you're looking for. Whereas if it's randomly dispersed mutations throughout the whole process, when you're dealing with like pseudogenes or viruses or oncology related, mutations, things like that, our probes start really breaking down around 10 to 15 base pairs. So we take that into account when we're ever doing, especially any design that's oncology based, to make sure, well, if there's only going to be like a mutation or two, our probe will handle it just fine. Or for virus strains or bacteria strains, things like that, we want to make sure to not make too many different probes for all the different strains. And if one probe can handle up to, let's say, 95% confidence and be able to pull out X amount of strains, then maybe you can have one probe to pull out 5, 10, 15 strains, whatever it is, to help save you money, where you're not having 10 probes pull out 10 different strains, especially if you're looking at one specific region of interest over those strains. So it really helps, save you money and cut down costs, and it helps, really know that our probes and efficiency is going to work really well, especially when you're dealing with like oncology or things like that. And kind of speaking of, this randomly dispersed and all that, but realistically, let's just say we're talking about one SNP, one single mutation. Well, very simple, very easy. You can just let us know, all right, here's the chromosome, here's the start and stop of that mutation. And then we'll design a probe, with the SNP over in the middle. But sometimes you do have these little issues where, well, let's say your SNP is near an ALU, a line, a sign, a repetitive region of the genome. We do take that into account when doing the designs, and we'll shift our probes accordingly, up or down, to make sure it will fit and to avoid any repetitive region in the genome, so you don't have high off target and issues with the said probe. So we do take this all into account, with our algorithm. And let's just say you want to maybe work with copy number variation. Well, all you need to do is provide us, the genes what regions of interest, what resolution you're trying to look at in doing the design, and then we can help kind of design the probes accordingly. Most of the time, if you're just kind of looking at our exome or just want to span across a gene, just doing our normal tiling, however we go about doing it, where we try to ensure that our probes completely span the exon without really going too much padding into the intron. This works perfectly fine, especially for just looking at exonic level or just gene level copy number. But sometimes people really just want to look at copy number across a specific region or a specific chromosome, or all across the whole gene, not necessarily just the exonic level. And then we can just space evenly across this gene or said region the probes, and not really care too much about just ensuring that you're still be able to pick up mutations or things like that in the exons. So kind of let us know. So anytime you have a copy number in there, we can take both approaches. And most of the time when you're doing like an oncology hotspot panel, this is perfectly fine for most people, just going with option one and just having our normal tiling, our normal way we design the probes, and then you just look across that gene of interest. And a lot of times we get a lot of questions for fusions, whether RNA or DNA based fusions. And in both cases, we kind of take into account, well, especially for RNA, give us the FASTA sequence or the fusion transcript ID. Let us know what is the actual fusion event. If you're looking at DNA and you don't have any idea, then we'll just tile across, add probes over across the intronic region, the exonic region, and then just, have many probes there to hopefully pull out and enrich the random chimeric events that occur. Whereas with RNA, sometimes you can just be more specific and say, I just want this exon to that exon, or just provide us the sequence, however it is. And then we go about designing then the probes accordingly, where we could combine, if you just give us this exonic region and this exonic region, we can combine the sequence together, make a hybrid fusion ourselves, or you can provide it to us, like I mentioned. We usually extend about 240 base pairs up and downstream, just to be able to add more probes there, to be able to ensure that you're going to be able to pick up this novel fusion or possible novel fusions that could actually span your said known fusion as well. We can make a mutant probe just for the known fusion, or we can just, again, add many different probes across this extended region to be able to then hopefully capture any novel fusions that could occur. And for a long read side of things, so we recently came out with a joint procedure protocol with PacBio. And so we have a good collaboration with them to be able to do long read capture. And that just entails using our mechanical frag library prep kit and our adapters and going through our hybridization with a little bit of minor tweaks. And then you go into the PacBio, Smart Bell process and then put it on the PacBio system. Same concept holds true if you want to do for Oxford Nanopore as well. The only problem is sometimes with Oxford Nanopore, the quality could be a little bit poor with the fact that our our indexes are 10 base pairs on each side, are dual indexes. So you have 20 base pair dual indexes, such that the 20 base pairs maybe isn't enough to really resolve each sample. So you might not want to really multiplex into the hives, depending on how you want to go about doing it. But something we can always talk about later if need be. But with the way we go about tiling for our long read is that we found you want to do lower spacing. So everything I was mentioning earlier was utilizing 1x tiling, essentially just one probe covering whatever the region is and trying to make sure you overlap them. But with long read, we want to make sure it's sparse tiling because you have longer read fragments. You have these fragments are about 5 kb, 3 kb, 10 kb, whatever the size is. The more of these biotinolated probes that you add across this bigger piece of the DNA, it could break the DNA. So what we found is you don't really want to 1x tile most of the time. You want to avoid that and be able to then tile more sparsely, whereas this probe actually works pretty well to then pull out these bigger regions of interest that you're looking for and not put too much strain on the piece of DNA with the biotinolated probes there. So most of the time, we go with 0.25. We can go even more sparse with 0.1. We can go a little bit more if we need to, tiling as well with 0.5. Up to you, but we're happy to work with you in whatever you need. But just know that we do have long read capture as well, if need be. And one of the biggest offerings that we has seen a big uptick in is actually our methylation offering as well. And like I mentioned with our methylome. So way we go about doing it is we design against our probes against the converted DNA, such that we assume you make a library prep, and then using our library prep kit, you actually then convert the unmethylated Cs to uracils. And through that process, you're going to have a diversity there across the whole. When you go to capture it, you have a lot of diversity where you now have Cs that turn to essentially uracils, that eventually Ts once you do the PCR, and the methylated Cs stay Cs. So you have all this diversity. You have these DMRs. You have many options where the Cs could be Ts or it could still say Cs. Well, that's where we then just throw a ton of probes at it and then rely on our probe mismatch tolerance, like I mentioned earlier, to help pick up all the varied degrees of methylation across that region of interest. So we design against fully unmethylated, fully methylated, and assume that. We then take both the sense and the antisense in both scenarios, and design using our double-stranded probes, a set of probes against each one of these sense and antisense strands, such that you have 8 possibilities of probes across this 120 base pair region that you're looking at. And so it's pretty cool. Throw a lot of probes at it, and you get this really good being able to capture the DMRs across, regardless if it's 0 or 100%, 25, 50% methylated, whatever it is, you can capture it to a really good degree. Something, I can go into more detail later as well if need be. So just kind of closing up. So what makes Twist better than the competition out there? One of the biggest things is that we use double-stranded DNA probes, and 100% of our probes are biotinnylated, so you have really good high capture efficiency. One of the biggest things that I kind of didn't mention across the whole thing, my presentation, is though we strive for exceptional uniformity. We want to ensure that your captures, however we go about designing, whatever the custom it is, it's going to be pretty uniform whenever you're going to go sequence it, such that it helps save you money then on the sequencing costs if you don't have to deal with over or under sequencing specific regions and all that. And one of the other biggest things too is we NGS QC all our final probe libraries, such that we ensure that our balancing of our probes and the sequences are exactly what we expected them to be before we provide you the probes, so that you're not getting any issues when you actually go about doing the testing. And the kind of, like I mentioned throughout my talk, is we can customize however you want. We have the scalable, small, large panels, things on those lines. We have whatever kit you need, whatever, we can do long read, short read, we can work with you. And at the end of the day, our striving is to help save you money on the sequencing costs in the end when you're doing these capture panels. And that's all I have to, talk about today. Here's kind of the sales reps. If you ever want to reach out to set up another meeting with me, Sarah covers the NGS side of things, and Brennan, he covers the synbio side. So I can take any questions now.
Awesome. Thank you. Yeah. If anybody has any questions, put them in the chat, the QA box. I got a question to start with though. So you did mention using the probes for enrichment for like 10x sequencing and things like that, which is awesome. But have you also used the probes for depletion with like a 10x workflow to remove things like ribosomal content or kind of unwanted genes, that way you're really capturing things that are more of interest?
That's a great question. I actually had a crazy idea eventually of using like our exome probes to deplete for human than to go into bacteria. That's a completely different story for like microbiome. But for RNA side of things, more often than not, people just use it to enrich, less deplete. I guess you could do it, but more often than not, people just care about, well, I only care about these transcripts, so I rather just sequence them and not worry about the background, as opposed to trying to pull out the background and see what's left.
Okay. Cool. Anybody else with questions?
So I had a quick question. So this is with the HLA kit that you mentioned. So is this for the long reads?
Yeah, so both, actually.
Both, okay.
So, yep, both a short read and a long read option for that.
All right.
And even, I didn't kind of mention it, I guess it kind of went in passing a little bit, like with everything else. With our long read, we can even do like, iso-seq as well, capture too. So you can have long read transcripts and we can still pull those out, as well. And I have a couple of clients doing that.
Cool. Okay. Anybody else? Okay. Well, if anybody does come up with any questions later, obviously feel free to reach out to myself, the Genomics Center, or Alan and the Twist team directly. But thank you very much for an awesome presentation. We'll be getting this up, hosted for anybody who missed it, so you can watch it later. And I guess until next time, everyone have a great day, and we'll see everyone next time for nanopore sequencing. So thanks, everybody.
Take care.